much for the invitation to talk to you here tonight. I'm absolutely delighted to have an opportunity to share some thoughts on the journey along my career. As of the 1st of January this year, I've been on staff in UCC for 30 years, and the opportunity to talk to you about some of the issues I've learned along the way was actually a really welcome invitation. So I'm delighted you asked, and I hope you find some of the thoughts I'm going to share with you some way interesting. I suppose I've titled this The Molecule Making Journey. I'm an organic chemist, and no matter what else I do, the first thing in my heart is organic chemistry. So making molecules is what makes me tick, okay? So I was thinking back to the very early days, how I got involved in science and research. There weren't any scientists in my family, right? So, it wasn't that I was exposed to careers in research. And when I was 14, somebody somewhere decided it would be a good idea if Anita Maguire became a doctor. So the decision was made that I would take physics, chemistry, and biology for the leaving cert, because that was a good right thing to do, to go and study medicine. And I suppose what nobody had factored in was that I met a remarkably inspirational teacher, Sean Foley, pictured here. I was actually in the first class he taught when he graduated from UCC, and he sparked an absolute lifelong passion for chemistry, which has lasted to this day. I owe him a tremendous amount. And some of you may be aware of him. He actually sparked a lot of very successful young scientist projects throughout his teaching career, but he's a really remarkable teacher. But there's a lovely end to it. I was in the first class he taught chemistry to. My son was in the last class he taught chemistry to before he retired. But a lot of my passion for chemistry comes from Sean Foley's inspiration. And after taking up chemistry, I decided medicine wasn't for me, and I pursued a degree in chemistry, and it was probably the best decision I ever made in retrospect. And then I came into UCC to study chemistry. So I was in the class that graduated in 1985. And this year on the steps of the Kane Science Building was my class of chemistry students. And many of the people in this photo here today are still the people I would count as my best friends. That's me there with the really bad green sweatshirt. But a tremendous group of people. And the staff in chemistry at that time were a really inspiring group of people. And the Kane Building wasn't quite as old then as it is now, but it was a really positive step in my career. And throughout my undergraduate degree, I actually had an opportunity to get involved in research along the way. So in the summers after my first year and after my second year, I undertook a summer research student in the laboratory led by Professor Tony McCurvey. He was the professor of organic chemistry in UCC at the time, and arguably I would say the greatest organic chemist Ireland has ever seen. But he took me into his research lab as a first year and the second year student, and I worked with the PhD students in the labs. And this was my first contact with research in synthetic organic chemistry. And it's true to say I got addicted, and my career has developed since that time. And then so I spent 2 summers in the labs in UCC doing research. And then the following summer, when I'd finished third year, I decided to get some experience in industry. And I worked in Penn Chemicals in Corribini, which was my first exposure to large-scale manufacture of active pharmaceuticals. And the pharmaceutical industry in Ireland is one of our most important sectors. And at that time, to go out and experience firsthand what it was like to work in industry was a really exciting experience for me. And the experience I gained over those 3 months into working in the highly regulated environment of the pharmaceutical industry has paid off many, many times over. And that site, what was Penn Chemicals in Corribini, later became the GSK site in Corribini, and now actually in the last year, it's been taken over by Thermo Fisher. But I got a real grounding in large-scale manufacture of active pharmaceutical ingredients and all of the regulatory aspects associated with pharmaceutical manufacture. And then I came back to UCC for my final year and undertook a final year project again in Tony McCurvey's lab. And I think because of the experience I'd had in industry, I actually managed to do a piece of research during my final year project, which was published in a paper. I think the experience I'd had in industry really meant that I was prepared to hit the ground running. So my first paper came out from work I did in my final year project. And then following my graduation, the summer after graduation, I decided it was time to spread my wings a little bit further. And I went to Glaxo Group Research in Greenford in London, right? So Glaxo would later become part of GSK, but at the time it was Glaxo Glaxo group research. And this was one of the largest groups of research chemists anywhere in the world, looking at medicinal chemistry and the discovery of new drugs. So it was really an opportunity for me to gain experience in a totally different arm of the pharmaceutical industry. So, you know, the summer after third year, I'd experience in manufacturing. Here I was immersed in the drug discovery and the medicinal aspects of the pharmaceutical industry. And those 2-3 month periods have been, without any doubt, the most important 2-3 month periods of my career, because I've built on the experience I gained at those times, and I've used it in so many ways throughout my career. The man I worked with, the person who was in charge of medicinal chemistry in Glaxo at that time, was Stan Roberts. And you'll see he'll pop back up again later on in this presentation. But right back there, 1985 was a really exciting time in the pharmaceutical industry. There was a feeling at that time that with the developments in computer science, that we'd be able to predict which compounds we'd need to make for drugs, and all the organic chemistry you'd simply be able to do the work on a computer, and the organic chemist would ultimately make the right answer. Here we are, some 35 years later, and that still hasn't come to fruition. But you know, the belief in the changes that were happening at the time were really, really exciting. So I had a tremendous experience there, and you know, it paid off to see the pharmaceutical industry from a very different perspective. Then I came back to Cork to do my PhD, and I worked with Tony McCurvey, who was absolutely a tremendous mentor. You know, you couldn't have asked for a better supervisor for a PhD. We all held him in such high regard. So I spent 3 years undertaking research in UCC for a PhD, and here's the title of my thesis: Synthetic and Stereochemical Aspects of Intramolecular Reactions of Alpha Diazoketones Catalyzed by Rhodium Carboxylates. And again, that PhD was partly, I was supported by Glaxo, you know, the forerunner to GSK, to do that research project, it offered me fantastic opportunities, because we got to visit the research chemists in Glaxo over the course of my PhD, and it meant that I had continued contact with industry over that period of time. But the reality was the day-to-day interaction with Tony as a mentor was absolutely pivotal in my research career. I would still identify him as the best mentor I've had throughout my career. And if you actually go into the university library, you'll see my PhD is there. It was one of the PhDs digitized about 5 years ago when they digitized the historical stuff. So it's there for posterity. I wouldn't advise anyone to look at it at this point, but it's there. And what was I doing, right? So the whole central element of my PhD was focused on chirality. Organic compounds exist. If they don't have a plane of symmetry, if they have a plane of symmetry, they're achiral. If they don't have a plane or a center of symmetry, they're chiral molecules. And what that means is that they have a sense of handedness, right? So you can have a left-handed molecule and a right-handed molecule. And we call this a stereocenter here. But the groups around the tetrahedral carbon can be oriented in 2 different ways, so that the 3-dimensional orientation in space is different. And as a result, the compounds form non-superimposable mirror images or enantiomers. And we've known this for quite a long time. Pasteur would have identified this well over 100 years ago. But really, the importance of it came into its own in about the last 30 or 40 years. And here you can see why. So here you've got 2 molecules. These 2 here are mirror images of one another. You can see each of the different groups. And if you have a mirror down the center, you'll see they reflect onto one another. When you turn this here, you see nothing you could do will enable you to superimpose this one onto this. You can superimpose the red and the blue, but then the green and the black are in the wrong place. And in a normal laboratory environment, that chirality makes little difference. If you think, when you pick up a book with your left hand and with your right hand, it behaves exactly the same. So when you put molecules which have this sense of handedness into a symmetrical environment, it doesn't matter at all. But you know that if you take your left hand and try and put it into a right glove, things go wrong very quickly. And in a living system, all of the chemistry in our own bodies have a sense of handedness themselves. So when we use organic compounds as drugs, if you put the left-handed drug into the system, it may work very well. The other hand will have a completely different property in a living system. And we've known this for about 30, 35 years. But the technology to actually produce the compounds as single enantiomers lags way behind the importance of actually delivering it. So my research project was focused entirely on this problem of how do you make compounds as single enantiomers. And the example which is often quoted is the case of thalidomide. Thalidomide was developed in the 50s and 60s, and it was used. It was given to pregnant women for morning sickness. And this enantiomer, this mirror image form of thalidomide, was considered quite safe. And then it was this enantiomer of it here which caused the very severe deformities which resulted in children being born without limbs. In fact, it's a really bad example because in a living system, 2 enantiomers of thalidomide can interconvert, but you'll often hear it quoted. But in the pharmaceutical industry, you'll hear the term chiral switch, where many of the companies take existing compounds which are on the market as racemic mixtures, and they generate new single enantiomer versions. And because you no longer have the unwanted inactive enantiomer, the safety profile associated with single enantiomer drugs is much better. Because you can imagine at the best case scenario, the second enantiomer will simply sit there and be inactive, and you're simply taking twice as much compound as you need to. But in the worst case scenario, you'd have undesirable side effects. So it's really important. Any drug now which goes through to the FDA for approval will essentially be required to be in single enantiomer form, unless there's a very good reason why it isn't. But our technology to actually produce the compounds in single enantiomer form is still a real challenge, right? So what I did during my PhD was this simple reaction here. We took alpha diazosulfones of this type here, where we have a nitrogen group adjacent to a carbonyl and a sulfone, and we exposed them to a rhodium catalyst. So this here is the rhodium catalyst with some carboxylate units around it. And what happens here is that the rhodium knocks the nitrogen off this here, generates a carbene, and then it undergoes a really selective CH insertion reaction into this position. So the rhodium, if you like, delivers that carbon in and inserts it between this CH bond. And the impact of that is that you form a new carbon-carbon bond. And this reaction had been developed about 5 years before I started my PhD. But the novelty we brought to it was we developed for the first time asymmetric catalysts where we put chiral groups here on the rhodium, and we controlled which face of this CH bond we'd insert into. So we controlled the stereochemistry at this center. And this was the first example in the literature of rhodium catalyzed transformation of diazo compounds with enantiomer control. And so it sparked 30 years of research, which continues today. But they were really fun days. And these are the papers that came out of my PhD. This is the one that describes that first example of asymmetric synthesis. And that paper continues to be cited today by many researchers all over the world. It's one of the most important papers I've published throughout my PhD. And then after I finished up in December 1988, and I headed off for a postdoc. But I came back in 1989 for the graduation, and you can see here my mother interrogating Professor McCurvey about the details of the chemistry I'd undertaken. So it was a great celebration of the time in UCC. And what did I do on graduation? Well, the first thing I did was I decided to go overseas and get some more experience in research. If you want to develop a research career, you need to get experience in different labs, and you need to be exposed to different approaches. So I went to work in the laboratory of Alan Krief in Namur, here's Namur, in the French-speaking part of Belgium. Okay, and I thought, I had my Leaving Cert French. I thought I was absolutely fine in French. I didn't realize there was a big difference between being able to write your Leaving Cert paper and actually being able to immerse yourself in a laboratory where you're the only non-French speaking person. So I learned a lot of French in my year in Namur, and I also learned an enormous amount about what happens in Brussels. I had one of these scholarships through the European Commission, which meant I spent some time in Brussels, and it gave me an insight into the European Commission and how they operate around research in Brussels. And it was a fantastic experience. Of course, I also took advantage of the fact that you could take a train to anywhere in Europe, so I traveled around quite a lot, but it was a really positive experience. And then after that, I moved back to the UK. I worked in the University of Exeter. And here, those of you who are focused will recognize, here's Stan Roberts again. In the interim period, Stan had left Glaxo Research and had gone back to university research, and he was the professor of organic chemistry in the University of Exeter. So I joined his research team. And at the time, this was at the height of the early days in terms of discovering drugs for HIV. And I was employed on the MRC AIDS-directed program to work on some novel compounds with potential activity against HIV. And this was the molecule which was the target of my work while I was working with Stan Roberts. And this was a really tremendous experience. Dan left me deal with the MRC myself. So I went to all the meetings. I met all of the biologists. I met all of the people involved in running the program. And it was a tremendous learning curve for me because I hadn't studied biochemistry or biology at third level. So, the way my exposure to biology had stopped at leaving cert level, and suddenly being immersed in this world where actually 90% of the researchers were not organic chemists was a real learning curve for me. And what I was trying to do was, this compound here is a really unusual compound, nucleoside, and it's one of the few naturally occurring fluorine-containing molecules which exist in nature. And we wanted to explore this as a lead for a whole series of new compounds against HIV. And it was a challenging year because it turned out the bug that made nucleosidin had forgotten how to make it, okay? So it was originally discovered in 1956, and in the interim period, the bug had mutated, so it stopped making it. So we were not able to derive it from the bug, but what we did instead was that I actually developed synthetic routes to this, and I developed a lifelong interest in nucleoside chemistry, which has paid off in the long run. But that was a really exciting year. And then in January 1991, an opportunity came up to come back to UCC as a college lecturer. In fact, my PhD mentor had moved, and therefore there was an opportunity to come back and develop an academic career of my own. And for me at the time, the really attractive point was to be able to come back and drive an independent research group of my own. And I remember walking in the door in January 1991, and I thought there was two elements to being an organic chemist, to being an academic. One was obviously the teaching and learning, the process of developing programs and teaching the students, and the other was research. And I was passionate about each of these two areas in the career. But what I didn't realize that day on the 1st of January 1991, that there have been two other elements to my career which have been just as important. The first one is interacting with industry. And this has become a huge part of my career, leveraging the insight I gained from the two placements. And the second one is engaging in the development of research and innovation policy at a national level. And I happily set off on a 30-year journey, engaging in all four of these different pillars. And indeed, these are not distinct pillars, they're all overlapping. So, my work in teaching and learning has played into my work in research and work with industry and my work at a policy level. And I would say I've spent the happiest 30 years working in each of these elements in various different ways over the interim period. And in 2002, I was appointed as an associate professor of organic chemistry. So what did I do over this period of time? Obviously, I taught a lot of students organic chemistry, but I suppose some of the innovations that I introduced was that I was the person who drove, prior to my time in UCC, chemistry in UCC focused very much on traditional organic chemistry, but based on the exposure I had, particularly in the UK, in the time I'd spent in Glaxo in the medicinal chemistry group, I recognized that it was important that our students were also introduced to medicinal and pharmaceutical chemistry, particularly because the pharmaceutical industry was such an important and growing sector in Ireland. So we introduced the teaching of medicinal and pharmaceutical chemistry for the curriculum for the first time. And we developed the degree program, and it's now known as the CPC degree program, the chemistry of pharmaceutical compounds. And this program was unique. We introduced it in 1998. We built into it work placement in industry so that every student undertakes a placement in industry. And we brought people in from the industry to contribute to the program. And at the time, that was groundbreaking in terms of the approach to teaching within chemistry. And that program has continued to attract top quality students ever since. But the key feature here was that prior to the 1990, chemistry and biology were two really distinct fields of learning, right? With chemistry, we could know everything about a molecule. We knew all about its structure. We knew it in detail. In biology, the ability to study the molecules at detailed level was really constrained until about 1980, 1990 time, when the technology actually enabled us to study large biological molecules, proteins, membranes, enzymes, at the same level of molecular detail that we could study them in organic chemistry. So around about this time, around about the 90s, it became possible to bring together the disciplines of chemistry and biology. And the CPC program was actually the first attempt we had in bringing together those disciplines and enabling students to learn at the interface between chemistry and biology. So that was a really significant early step in my career. And where that led to afterwards was in 2003, UCC introduced a School of Pharmacy for the first time. Prior to this, the only school in pharmacy in Ireland was in Trinity. But because of the work I had done in developing the CPC degree, when the university decided to look into the development of School of Pharmacy, I became centrally involved in the development of that, and the making the case for it at a national level. And then once we'd secured the school, actually becoming involved in the startup of the school, the development of the programs, hiring in the staff. And this was a really exciting part of my career, creating a school in a university from scratch, where you're not constrained by any preconceived ideas, is a really exciting thing to do. And you'll probably be aware of the pharmacy building here. So I was centrally involved in the design and the development of that building. I could still draw the map of the building. And this again was a really exciting period in my career, being involved in something so new and starting from scratch. It was a very exciting time. So I was appointed as the first chair of pharmaceutical chemistry in 2004, and that's the position I've held ever since. But my chair actually spans the School of Pharmacy and the School of Chemistry, so I'm a professor in both of them. And as one of them is in SEPS and one is in Medicine and Health, it's given me a very unique position and a unique view of the university. But the startup of the School of Pharmacy and the development of the building was really a highlight in my career. And again, there was a lot of luck involved in this, right? Because I remember just before Christmas in December 2001, we got the phone call that we'd been approved for the School of Pharmacy after about an 18-month process to get to that point. And in exactly the same week, we got the call that we had been awarded funding from the HEA under a program called Programme for Third Level Research, PRTLI 3, that I had secured 7 million for a research facility called the Analytical and Biological Chemistry Research Facility. So serendipitously, those two announcements came the same week. And what it enabled us to do, we took the 7 million that we got for the ABCRF, and we took the 6.5 million we got for the School of Pharmacy. We put them together, and as a result of the timing, we were able to develop a really state-of-the-art building in the pharmacy building. If there'd even been a 6-month delay between them, the planning for the building would have gone ahead and we wouldn't have been able to combine the two. So just the timing on the two of these was a real stroke of luck, right? And what the ABCRF was about, it's exactly the same as I mentioned to you in terms of teaching. It's about bringing the research, which is at the interface between chemistry and biology, and bringing all of those people together to work in an interdisciplinary centre so that we can actually deliver the full scope of activity at this interface. So I've been director of the ABCRF since 2002, and I have a tremendous set of colleagues, obviously a lot of people involved in organic chemistry, pharmaceutical and medicinal chemistry, analytical chemistry, but also colleagues from biochemistry and bioanalysis, bioinorganic chemistry, and then people from the pharmaceutics in the School of Pharmacy. So there's a great team of people, each bring their own expertise to address very challenging issues at the interface between chemistry and biology. And one of the key features is when we reach out to work with the pharmaceutical industry, we have all the disciplines together internally, so we can reach out and work on collaborative projects with industry in an interdisciplinary sense. And this, when you walk into the pharmacy building, the top floor on one side is the ABCRF and on the other side are the labs in pharmaceutical chemistry and the School of Pharmacy. But the fact that we got the two at the same time meant that we have a really tremendous research suite as a result of that. So again, that was a high point in the career. And through the ABCRF, we have a number of people who work in the pharma industries in the region who are adjunct professors in the university. For example, Pat O'Neill from Pfizer is associated with us, Andy Whitehead from GSK, and they come in on Fridays and they participate in the postgraduate seminars and they engage in the research life of the team. So, we're very fortunate to be close to some of the leading companies in the world, and this enriches the experience of the research teams to an enormous extent. So that was a really exciting part of the career. And how did that play on, right? So it's about 2008, the country decided to invest in a number of research centres really focused on conducting research at the highest level and working at the interface with industry. And because of the work I had done as director of the ABCRF and in partnering with industry, I became involved as one of the co-PIs and one of the leads in a centre called the Synthesis and Solid State Pharmaceutical Centre. It's now just known as SSPC. The name has really been abbreviated. It's laid out of the University of Limerick, but I lead lead the molecule strand. So I lead the synthetic chemistry strand together with my colleague Pat Gyrey from UCD. And we became involved in this in 2008. And, you know, the work I'd done with the ABCRF within UCC led to being involved in this at a higher level. And the SSPC now brings together all of the universities in Ireland working at the interface with the pharmaceutical industry and all of the leading companies in Ireland and internationally. So this is a very large, there's probably about 350 people now involved in SSPC, and it works across all different elements. So the part of it that I work on is the synthetic chemistry element, but it's a really positive experience. And I would say, you know, we progressed to this from the ABCRF and we continue to work within the SSPC. So it started off in about 2008 as a 7 million centre. The next round of it from 2013 to 2019 was a 40 million euro centre, and now it's restarted the next round of it in 2019, and it's now a 61 million euro centre. 80 PhD students, 9 universities, and 25 industry partners. So every quarter we get together as a group and share our experiences, and it's a really exciting environment to work in as a synthetic organic chemist. So what do I do? All right. So I want to give you a little bit of a flavour of my research and the kind of projects which are underway in my research lab. So we're active in a number of different areas. I never believe in confining my interest to a narrow area, but they can generally be classified as asymmetric synthesis. You know, the bug I got in how you make left and right-handed molecules has never left me. Okay, so, you know, we've continued to be very active in that area. We're also very active in new synthetic methods, you know, finding new ways of making new compounds, not just another way of doing the same thing, but reaching outside the box and finding new ways of doing chemistry. And we've an active strand in the development of new medicinal compounds, building on the work I did with Stan Roberts in Exeter. We're also involved in crystal engineering in collaboration with Simon Lawrence, and we've got an active program in continuous flow chemistry. And you see here some of the molecules we've worked on over the years. So I'll just give you a small flavour of some of this chemistry. So for much of my career, I've been very involved in exploring the chemistry of alpha diazocarbonyl compounds. These are really versatile synthetic intermediates. You simply, when you remove the nitrogen from here, because of the very high stability of nitrogen, you get a very reactive species, and it can do some transformations which are really very different to other types of chemistry. This one here, in terms of forming 3-membered rings, this is the best known way of generating highly strained 3-membered rings. But also they add on to aromatic rings, and this is the mildest way known to mankind of destroying aromatic systems. So there's a lot of powerful transformations that diazocarbonyl compounds undergo. But the one that I've been most interested in over my career is the whole area of CH insertion, where you insert the diazocarbon into a CH bond and form a new carbon-carbon bond. And over the last decade in particular, we've been very actively involved in exploring the use of copper catalysts for affecting this CH insertion. So you remove the nitrogen from here, the copper atom sits here, and then it steers that carbon into this unactivated CH bond. And as a result, you form a new carbon-carbon bond here. And obviously the goal is to control the stereochemistry at this position. And the reason this is such a powerful transformation is that this CH bond here is completely unactivated. It's remote from anything else in the molecule. It's not active in any sense. So you're using a metal catalyst to actually pick one CH bond out of a whole long chain of them, activate that to react, and then do it with control of stereochemistry. So it's a really powerful transformation. And we've been very fortunate over the last decade to have made some very exciting discoveries in this area. And what you can see here is we've explored what happens if you vary the substituent at this position, or if you vary the ligands that you wrap around the copper. And what you see here is what gives you the best control of stereochemistry, with the best result here, around about 90% of one single enantiomer. And what's really important about this, the earlier work I'd shown you in this area had all used rhodium or ruthenium, precious metals, which are expensive and they're in limited supply. Being able to do this type of powerful chemistry with copper is a major advantage, because copper is much more readily available. It's much cheaper to access. But even more so, if you wanted to use this type of insertion process to make a pharmaceutical, then if you use copper to do the chemistry, and if there's a tiny residue of copper in the drug, that's not a risk to people who take it, because we have copper in our living systems. We require a certain amount of copper within our systems to work. So the risk is much lower. If you use rhodium or ruthenium, you've got to go to much more extreme lengths to ensure that there's absolutely none of the metal left within the drug. And of course, course, now because rhodium is in such demand for catalytic converters in cars, its availability, it's one of the most expensive precious metals now. So being able to move this chemistry to copper was a huge step forward. And many people around the world had tried this without success. The magic ingredient was we added in this little guy called sodium barf. And someday I'd be quite happy to tell some people more about this story. But we were the first in the world to show that you could do copper-mediated CH insertion with excellent control of the stereochemistry at this new stereo center. And over the years, we've developed this reaction. So we've shown you can form this carbon-carbon bond in lots of different systems. So or we can find 5-membered cyclopentanones, or indeed we can form lactams. And here you form beta-lactams, which are so important to the antibiotics. And you can form addition to aromatic systems. So this copper catalyst is broadly applicable across a wide range of transformations. And we can get outstanding enantioselectivities across each of these. So this has been a tremendously exciting project over the last decade, and it continues in our lab today. But one of the challenges we've always had when we talk to people in industry about our diazocarbonyl chemistry is that they glaze over because, of course, diazo compounds are hazardous, right? Anything which can release nitrogen is hazardous. And that's absolutely fine to do in a laboratory with 1 or 2 grams. But if you're in a pharmaceutical plant and you need to make a ton of a pharmaceutical compound, potentially having something release that much nitrogen over a short period of time is a huge hazard. And in particular, the precursors that are used to make these diazo compounds are extremely hazardous. So this is a reaction. It's a fantastic reaction developed by Regitz about 25 years ago, where you take a simple precursor and you react it with a sulfonyl azide. This one here, tosyl azide, is the best known example. And tosyl azide very effectively takes 2 of its nitrogens and transfers it to this position. It's a brilliant reaction. It works across a wide range of substrates and it's really efficient. The problem with it is tosyl azide has the same impact sensitivity as TNT. These are highly explosive compounds. So none of my colleagues in the pharmaceutical industry would relish the idea of having a ton of one of these in their warehouse. So, the ability to take this chemistry and do it at scale was always limited by that hazard. But something changed about 10 years ago, okay? And that was that you see here an announcement from the International Union of Pure and Applied Chemistry. They named in 2019 10 chemistry innovations that will change the world. And one of them was the concept of flow chemistry. So up until this time in industry, when they wanted to do a large-scale reaction to form a new compound for a pharmaceutical, they simply did it in a very large round-bottom flask. So it was the same type of process we do in a lab, but they simply scaled it up. And then the hazard associated with that was very substantial. The introduction of flow chemistry was the real first big step change in how we make pharmaceuticals at scale. And the whole idea of this is that when you want to make a compound at scale, instead of just making an enormous round bottom flask, what you do is you flow the reagents through pipes and you mix them together and you form them in flow. So you never have a large amount of any material, you generate it in flow and you generate the product and remove it. So this has become a major step change in the pharmaceutical industry over the last 5 years or so, with most of the major players becoming involved in this area. And we thought this was an ideal solution to our problem of diazochemistry. So none of my colleagues in industry wanted to use diazochemistry in scale because they didn't want to have a large amount of this hazardous sulfonyl azide route. And we said the introduction of flow chemistry was the ideal way to address this. So instead of making a large amount of this hazardous compound, what you do is you take an aqueous solution of sodium azide, which is perfectly easy to handle, a solution of sulfonyl chloride, you mix them together and you form this. And then as you form it, you bring it on, you react it in the next step and you form your diazo compound. So at any point, you only have a tiny amount of this compound present. But if you run it for long enough, you can get up to a ton of the product. And this gets away from all the risk associated with doing diazochemistry at scale. And over the last 5 years or so, we've really demonstrated that this methodology makes it feasible to start thinking about diazochemistry at scale. And it's changed the perspective people have on the accessibility of this chemistry at scale. And you see here, we've developed 4 distinct protocols for precisely different situations, but we've become the world experts on doing diazo transfer in flow over the last 5 years. And this is what it looks like. You see here, this was a really important paper we published in 2017. And the whole reason that this paper is important, it was the first time we demonstrated we can start off with a substrate here, a simple compound. We make our sulfonyl azide in flow, so we simply take an aqueous solution of sodium azide, we generate our sodium azide in flow, mix it with the substrate and react them together. We put in a quench then to kill any dangerous material at the end of it. We separate out the organic and the aqueous layers, and here we have a solution of our diazo compound. And without ever isolating that, we pass it immediately through to the next step. We trigger a Wolff rearrangement and we isolate the product. So what's happening here is we're getting the power of diazochemistry. We're transferring this simple precursor to this really complex product, getting all of the power of diazocarbonyl chemistry. But at any point, you've got about 0.5 gram of the sulfonyl azide present, and you've got about 0.5 gram of this present. But you can transform through kilos of this by simply running it for a long period of time. And this was the first time anyone had demonstrated you could get the power of synthetic chemistry involving diazocarbonyls without ever having to isolate or handle either the dangerous precursor or the diazo compound itself. So this paper was a real step change in terms of the acceptability of diazocarbonyl chemistry in flow. And what we're doing here is that we're actually mixing the precursors to make the sulfonyl azide. Then we bring it in to do the reaction, and here's the coil. We simply run the tube around in a coil to give it long enough to react. Then we add in this quench, which kills any of this which is left behind. And then we end up with quite a complex mixture. So we've got our product, we've got all of these other species in there as well. So there's a full mole of the sulfonamide, because when this hands over its nitrogen, you're still left with one nitrogen. There's a quench, there's any unreacted starting material, there's the triethylamine. So it's a really complex mixture. And if you want to go on and do chemistry with this, you don't want to be bringing all these other guys with you. And this was the magic. We designed a phase separator so we could separate out the organic layer, which contained our design project, and everything else went out in the aqueous phase. And we were able to separate this in flow. We didn't put it into a separating funnel. We could separate them out as it was formed. And what you end up with then is a clean solution of the alpha diazocarbonyl compound, and we brought that into the next reaction. So conceptually, this was a really important step forward in diazocarbonyl chemistry. And this is what it looks like in practice. So up here, you have the bottles of all the reagents. Here's the first set of coils where we're doing the diazo transfer. So they're simply running around in tubes for long enough to react together. Okay. Here then we feed it into the separator, and this is what the separator looked like. We designed it so the aqueous and the organic layer would separate. The organic layer comes off the top, the aqueous layer comes out the bottom, and they separate out here, and then we bring it into this coil for the subsequent reaction, and we collected the products. And this is what continuous flow chemistry looks like in reality. But we've had tremendous response all over the world, in industry and in the academic world, to the work we've done in this area. And what I'd like to think now is that when people are sitting down to think about the routes they use to synthesize new pharmaceuticals, that at least they consider diazochemistry as an option, because we've shown that it can be handled safely. And here's another example. These were a class of diazo compounds which we'd worked with for a very long period of time. Stuart Collins, actually, as part of his PhD, some of you may know him, he did some of this work, and these are really unstable compounds. They're really challenging to make. You know, on a good day, we'd get a 30% yield of one of these. And the problem here is that these compounds are not very stable, so they're already decomposing in the reaction mixture. And here we found a trick with continuous flow chemistry, whereby instead of having the base in solution, we immobilize the base and we flow the reagents over it. So we left the reagents in contact with the base for long enough to transfer the diazo group, but the minute it was done, we washed them away from the base so you don't get any decomposition. And as you can see here, we get an immediate jump in yield from a 28% yield up to 88% by doing this in continuous flow. And we never anticipated this power beforehand. But even more dramatic, here's the reaction when you do it in flow. Look, a lovely clean yellow solution of the diazo sulfoxides. Here's the same reaction if you do it in a traditional flask where you're stirring it and the compound has time to decompose. So really dramatic improvements in chemistry through doing the work in continuous flow. And our team continues to work in this area. And then finally, I just wanted to give you one last example. And this is some work we do in the medicinal chemistry area. And this goes back to the time I spent with Stan Roberts in Exeter working on nucleoside chemistry. And really, we designed this novel series of compounds compounds. They're called alpha carboxy nucleoside phosphonates. So they're built on a nucleoside. They've got a phosphonate unit attached to it. But the key here, the magic here, the unique feature is we attach the carboxylic acid on adjacent to the phosphonates. These are the first classes of this compound described in the literature. And what we found was that these compounds here are just as active against the key enzyme in HIV reverse transcriptase, as the compounds which are clinically used for treatment of HIV. They're really active, but the more important feature is the mechanism by which they act. The way in which they inhibit HIV is completely different to any of the other compounds which have been investigated. And as you may know, HIV undergoes mutation, so there's a constant need to find new ways to attack HIV as it mutates. So therefore, discovering, after 30 years of HIV research, to discover a new set of compounds with a new mode of action was really exciting for us. And this is how it works. So our new CNPs, they mimic the natural triphosphate, which is used as the building block of DNA. And by doing that, they inhibit the enzyme. And this here was the cover of JOC when that research was published. And here you see the actual crystal structure of our CNP, this compound here, sitting in the active site of reverse transcriptase of HIV. So around here, what you're looking at is the active site of HIV reverse transcriptase, which is the most important target against HIV. And you can see our compound sitting in there. You can see base pairing with the template and the enzyme. But here's our carboxyphosphonate. So here's the phosphonate, here's the carboxylic acid, and they're critically important in bringing that into that compound here. And what's happening? The way in which HIV reverse transcriptase works is it's the enzyme which generates, HIV brings its genetic information into the system as an RNA molecule. And before it can take over the machinery in our cells, it has to translate it into a DNA copy. So HIV reverse transcriptase is the enzyme which makes a DNA copy of the viral RNA. And what it does is it coordinates the building blocks. So here's a nucleoside triphosphate. They coordinate onto two magnesium metals in the active site of the enzyme, and this is how they're incorporated into the growing DNA chain. Our molecules, or alpha CNPs, sit in exactly the same way. They mimic this triphosphate, but you get a direct coordination of the alpha carboxy phosphate onto the metals, and it mimics it in exactly the same way, so you get base pairing here. And this had never been seen before. This is the first monophosphonate which mimics a triphosphate without having to be phosphorylated further. So it's a really exciting discovery in our team in the last decade. And once again, this goes back to our time of our left and right molecules. We assumed that the active enantiomer of this was the one that looks like the natural enantiomer, but when we actually checked it out, it's the unnatural enantiomer. It's the one that does not look like the natural nucleosides, which actually retains all of the activity here. So research continues to surprise you. No matter what you predict, you'll get unexpected outcomes. And this was the chemistry we used to synthesize this. I'm almost embarrassed. It took us about 15 man years to do this work. It was really difficult chemistry. Now that we've cracked it, we can actually do this from start to finish in about 2 or 3 weeks, but it was really challenging chemistry. And this here was the key step where we formed this oxygen carbon bond, tying in the phosphonate carboxylic acid. And again, our old friend rhodium was used here. But this was a really exciting journey for us, undertaken by very talented PhD students in the team. And I'd like to just highlight again, one of the most important features for us in this project was the opportunity to work with Jan Balzerini. He was a professor in KU Leuven, arguably the greatest university in Europe, and he had been active in HIV research for his whole career. But when he found these compounds, he jumped on a plane actually, and he came over to Cork. He was so excited, and he called these the molecules of the year. He's retired now for about 5 years, but he continues to collaborate with us because of his passion for these compounds, right? And just one final project I'd mention, this is in the area of biocatalysis, where we work with Jerry Reen and Fergal O'Gara in microbiology. And they, in their research team, they go out and they search in nature for novel enzymes. And one of the enzymes they found was a novel transaminase isolated from an organism in Lochine in West Cork. And we took that transaminase and we applied it to some really interesting chemistry. And what we've discovered is that not only could we control the stereochemistry at the site of reaction, but we could control the stereochemistry at a remote site as well. So one of the first examples of that. So there's lots of exciting chemistry we've been working on throughout the years. But in all cases, the interaction with other scientists who bring complementary skills is actually key key to success. So, and we'll continue to explore these areas. But in the last few minutes of my talk, I'd like to come back and talk about the work we've done at the interface with industry. I mentioned already just how important the pharmaceutical industry is to Ireland. And if you go into the IDA website, you'll see these stats. Ireland is the third largest exporter of pharmaceuticals globally in the world, right? Annual exports of about 80 billion. It's really important to the Irish economy. And many of you might be aware the financial hit to Ireland last year wasn't as bad as it might have been thought because the pharmaceutical sector did so well over last year. It really mitigated the effects. But 9 of the world's top 10 pharmaceutical companies are in Ireland, and Ireland is perceived as one of the best places in the world to scale up a new drug. And the reason why is that there's a collective knowledge within Ireland across all of the companies about how you do pharmaceutical scale up and how you do it in a way which is in compliance with the regulators, so the FDA and the HBRA. But the industry in Ireland is remarkable. 25% of all PhD graduates employed in industry are employed in the pharmaceutical sector. And the exports grew from 2012 from 24.5 billion up to 80 billion. It's a really important part of our economy as a country. And, working in Ireland as an organic chemist, there isn't a better place to work. And in particular in Cork. In Cork, we're surrounded by all of the large scale-up of small molecule synthesis in Ireland. There's another cluster around Dublin, but in fact, Cork has the largest cluster of small molecule scale-up in the world. So I describe our university as being the best place in the world to be an organic chemist because you're within a half an hour of Lilly, Pfizer, all the major players in this game. And over the years, I've been very aware that, the PhD graduates from our team, they're going out into the pharmaceutical industry and they're centrally involved in expanding the amount of research and development underway in the company. So while they're doing a PhD in our team, each one of them actually gets an opportunity to spend 3 months working as an intern in the pharmaceutical sector so that when they go out, they're ready to hit the ground running as part of a team. And here you just see 2 examples. So Rose, who's currently working in Lilly, and Valerie, who worked in MSD. But many of the people from the team take 3 months and go out and work in industry during their PhD studies. And we're fortunate to have such good companies to give them those placements. And here's a really great example. Dan Crowley just finished his PhD actually in December. But during his PhD, he went out for 3 months and he worked in the Pfizer site in Little Island. And he was working on flow chemistry in our lab. And this was they used him to actually do their first experiments in continuous flow on that site. And they actually won an award for the continuous flow hydrogenation proof of concept that he developed through that placement. And, for us, having those partnerships with the companies is a really important part of our work on a day-to-day basis. We've also had large-scale collaborations with some of the companies. One of the most exciting was a collaboration we undertook with Lilly, which ran for many years, from 2009 up to 2015. And you'll see here the postdocs who were involved in this project from our lab. We undertook 11 different projects and they were jointly led. So I supervised them as the academic supervisor, but then with chemists in Lilly and Kinsale and with chemists in Lilly in the US, all working together over a series of projects. And this was the first paper we published, which brought together our research lab, chemists in Kinsale and the chemists in Lilly in Indianapolis. And it got a lot of attention at the time. And over the period of that collaboration, there was a whole series of papers came out, and it was a very high profile collaboration linking the 3 research teams. So that was great fun. And at the time, we had an opportunity to collaborate with the NMR spectroscopists in Lilly. You see here the pigs flying, right? At the time, nobody ever thought you could link up a reaction to an NMR and look at the reaction in real time as it was happening. And we did a very exciting project with them where we took this long complex scheme we'd be looking at in our lab, and we actually looked at it real time in the NMR. You can see here's the starting materials, and as the reaction is proceeding, you can watch the formation of the new compounds as it goes through the cascade, and you can plot the disappearance of the starting material, the appearance of all the intermediates. So you can actually watch a reaction real time as it's happening. And 20 years ago, no one would have believed you could have done that, right? And this led to one of the most important important papers in the field. David had done his PhD with me, and then he'd gone out to Lilly as a postdoc, and he's now one of the leaders in this field internationally in using NMR in real time. And we now have a suite set up whereby we can do this real-time investigation of processes by NMR and by other technologies due to that work. So Prospect is probably the best suite in Europe to do that work at the moment. So that's all the research, right? Before I finish, I just want to give you some insight into the policy landscape work that I've been doing. So, throughout my career, I've always engaged in development of scientific policy at a national level. So starting off in the early years as a part of the Irish Council for Science, Technology and Innovation, and then moving on, I was a part of the Advisory Science Council, and then I was part of the Irish Research Council, which funds a lot of PhD students in the country. And indeed, I chaired it for a period of time. I'm currently on the board of NIBRT, which is the National Institute for the Biopharma Sector, and I'm currently the chair of the Research Integrity National Forum, which is the oversight body which ensures that people conduct research to the highest quality. So I was the inaugural chair for that, and I continue to lead it. But on a day-to-day basis, linking in with policymakers and ensuring they understand how researchers and industry work together has been a really positive element of my research career. And then I suppose finally, the other element I engaged in was academic leadership. I've been head of school in both chemistry and pharmacy, and probably I think I'm the only person in the university who's been head of two schools in two different colleges. One is in CEFS and one is in medicine and health. So it's given me a unique perspective on the university. And in 2019, I took over as Vice President of the Royal Irish Academy at a national level. And all of these roles in academic leadership prepared me really well for taking on the role as Vice President for Research and Innovation in 2011. So I spent the last 10 years of my career really in a remarkable position. I've had such a joy and an honor in leading out research in UCC, and it's allowed me to engage with many of these leading research institutes nationally. Some of the brightest and best researchers in the country work in these institutions, and it's been an honor to work with them throughout that 10-year journey. And throughout the time I've been Vice President for Research, I've been involved in, when I started, we brought in about 78 million euros a year of research funding. It's now over 100 million euros, and this has been a tough decade financially. So that was a really substantive growth. And in particular, we grew the amount of money that came from industry partners. So it's been a real pleasure. And I've really enjoyed leading out on many of the high profile elements of the research landscape in UCC. I've also been involved in all of the commercialization of our research, the patenting, spin out companies, licensing stuff to industry. So it's been a real blast. And today is actually my last day as Vice President for Research after 10 years. And, the honor to work with some of our leading researchers. These were just some recent highlights that have come in, where Jens Walter has pulled in a 3.7 million euro program to study disease. With 3 of our researchers attracted very prestigious ERC funding just at the end of last year, and some of our researchers have attracted some of the most high profile IRC awards. So really exciting time to lead research in UCC, and it's been an honor for me. But perhaps the most enjoyable part of it all is you'll be aware that last year, due to the pandemic, we had to close the research labs, for March until May of last year. There was little research underway in the campus, and then gradually we reopened the labs, and by September we were back up to full capacity. And for me, the process of reopening that and steering it through safely has actually been one of the most rewarding parts of being the Vice President for Research and Innovation. And we've been operating with between 11 and 1,200 people on campus since September without any outbreaks as a result of it. And then at the end of last year, we managed to secure 7.8 million to support the research teams which had been affected by the pandemic. And we were able to allocate this out to individual PhD students and postdocs whose research had been impacted. We were able to help 88 people last year. And here you could see the reactions. And we approved another batch of them just last week. So it's been a tremendously positive experience for me. But I want to just close out with the image of some of my research team over the years. What really gets me out of bed every morning is working with such talented researchers, each one of them adding in their own way to the richness of the chemistry we've undertaken. So 54 PhDs have graduated from the team, 42 postdocs. Most of them have gone out into the pharmaceutical industry. And I want to acknowledge in particular Stuart Collins, who co-leads the team with me now, and Debbie Curran, who actually makes everything tick in our research team. But it's all this group of people who really have made my career as an organic chemist worthwhile. And finally, I want to thank you for your attention.
Thank you so much, Anita, for such a fantastic STEMinar. I definitely am speaking on behalf of everyone here when I say, like, we've all learned so much from that STEMinar, and it was really fascinating to see how you've gotten to where you are today. So thank you so much. We're delighted to have you here tonight. So with that, I'm going to open up the floor to some questions. So if anyone has any questions for Professor Maguire, please feel free to send them into the chat, or else you can unmute your mic and ask them that way. Otherwise, I can start off and I'll ask a few questions as well that I have here. So I guess just to go back to the beginning, Anita, where would you say that your passion for chemistry stemmed from?
I think I was just really lucky that I came across a teacher who was very passionate, and he sparked off something that was innate in me, right? But I was really lucky. If I hadn't met him, if I hadn't had an inspiring teacher, I may have gone down a very different direction, right? So I think I was lucky. He set me on a journey where I really enjoyed it. And then I was lucky in having such a tremendous mentor for my PhD as well. I couldn't have asked for better. So I think people are really important in this journey as much as anything else.
Yeah, it's very clear the importance of having role models along the way. And what would you say helped you decide to become a lecturer after your degree?
Because I absolutely adore research. I just fell in love with the whole process of undertaking research. So for me, the idea of being able to drive a research program and share the love of chemistry with generations of students, I couldn't think of anything better, right? You know, for me, I don't work. I actually come in to do my hobby. So for me, it was a no-brainer. I loved it. I mean, the real challenge for me was at the time I left the country in 1988. It was a pretty grim time in Ireland. There was no new academic posts. I didn't think I'd come back. I thought I would never have the opportunity to come back. So it was a real surprise to have the opportunity to come back and to be able to drive a research career in Ireland.
That's great that, you know, that you can even say that, like, your work, you know, your job feels like a hobby because you're passionate about it. That's brilliant. And over the years, you know, what would you say is the biggest challenge you've encountered and how did you overcome it?
Yeah, the biggest challenge I've encountered is I get so enthusiastic about so many things. It's managing my time, right? It's juggling all of the areas because I love research and I love teaching and I love policy, and I have a family who like to see me occasionally as well. So the real challenge for me is how you juggle all of this and how you manage to keep the momentum going. But I've become very, very good at juggling and keeping things going. What I can do in a half an hour is remarkable. So that's always been the challenge, and also learning how to say no. You know, different people want different things from you, and being able to have the confidence to turn around and sometimes recognize that you actually don't have the capacity to do things. But for me, it's all about, you know, finding ways in which you can deliver everything you want to deliver, but still do everything to a very high quality.
Yeah, and like to add on that, then the opposite, what would you say would be your proudest achievement?
Well, my children come first, all right? But other than that, after my children, I would say my PhD students, the people out of the research team, are by far, I am so proud of what they've achieved. They've all gone out, they've developed exciting careers, they're fantastic people. So for me, the day I retire, what I will look back on is the people, and, you know, the way I've had played some part in developing a whole cohort of people who are going out and they're playing a really important role in increasing the amount of research and development within the industry in Ireland. So they're definitely my proudest achievement.
And I'd say they're very lucky to have you too as a professor. And I guess moving on from that, what would you say, you know, a typical day in the life as a professor of pharmaceutical chemistry looks like?
I'm glad you asked me that one, because there is no typical day. No 2 days are the same, right? So on any day, you could be involved in teaching, you could be involved in research, and normally it's a mix of the whole lot of them. So, you know, the way I might have a meeting with a PhD student in the morning, then I might give a lecture to a class, then I might have a meeting with one of our industry partners, then I might have a meeting with one of the policy groups at national level. And the real joy of an academic career is actually the variety of things you do, and the fact that you can shape and choose what elements of the role you want to focus on. So, you know, the typical day, actually, there is no typical day. And for me, it's always about how could I get more time to do more? I'm never bored. I'm never looking at the clock waiting to go home.
That's a very great way to be. You know, earlier on in your presentation, you mentioned that you had the great opportunity to go abroad and work and study. What would you like? Would you say you'd recommend graduates going out and pursuing postgrad programs abroad should the opportunity present itself?
I think traveling is really important. You know, I think particularly when you live in a relatively small country, I think the experience to go out and work in another environment is a really important one. And I think I would never have settled down in Ireland if I hadn't gone overseas and realized that actually what we have here is as good as anywhere, right? But I didn't see that before I left. I'm not sure it's essential to go overseas for postgraduate research. You know, lots of people would undertake postgraduate research in UCC and then spend maybe 3 or 6 months overseas to do some research during that course. I think it's important to take some time somewhere traveling at some point, right? But I don't think people should define, okay, I must go for this particular part of my career. When you're undertaking research, there's always opportunities to go and work in other labs. So it's about taking the opportunities as they come up and getting experience to work in different environments so that you're exposed to different approaches.
That's some great advice. Thank you, Anita. And just to, I guess, from your perspective as, you know, the VP of Research and Innovation, how do you think that innovation and research can be promoted and encouraged more on campus?
UCC is actually a very much a research-led institution, right? It's very much embedded in everything we do on a day-to-day basis. In terms of total research funding, UCC is usually number 1 or number 2 at a national level in terms of drawing down research funding. So research is very much a part of what's done on a day-to-day basis within the university. I think perhaps the visibility of it to the different cohorts, I'm not sure our undergraduate students are fully exposed as much as they might be to the research and innovation activities. And it's perhaps making more opportunities for people to be aware of just how exciting much of the work is underway within the institution and creating more opportunities for people to learn about it.
Yeah. And you mentioned also that you're involved with many national institutions, including the Royal Irish Academy and more. What do these roles involve?
Yeah, I suppose I'm Vice President of the Royal Irish Academy at the moment. And in particular, one of the roles I play for them is I chair the Diversity Committee. So, you know, we're looking at how we can ensure that, you know, the membership of a group is representative of the full diversity of society. And that's not easy to do in a traditional organisation. But also, you know, the Royal Irish Academy, literally, it's the place for the most highly accomplished academics across the country to come together and share their views, and they're very much involved in feeding into policy development at a national level. I think over the last year, we've seen more than ever just how much research comes to the fore in terms of advising national government policy. So the Royal Irish Academy would be one of the key areas of the country where those ideas are brought together and fed into the policy landscape. But it's very exciting to mix with people from different areas and different disciplines from all across the country. And the Royal Irish Academy is an all-island body, so it crosses the north and the south. So it's a great bunch of people, and it's a very exciting group to work with.
Yeah, it certainly sounds like a very interesting thing to be involved with. And as a concluding question, if there's no other questions that pop up in the meantime, so yesterday was International Women's Day, and UCC posted a lovely article featuring yourself and your advice that you would give to your 17-year-old self. Would you mind please sharing some of that advice with the audience here tonight?
Yeah, first of all, I was very embarrassed that that letter became public. iWish asked me to write it, so I wrote it for iWish. I didn't realize it would become as visible as it did, and ultimately. But I suppose some of the thoughts I shared in this, I remember when I came into UCC, I came under a lot of pressure to study medicine, and people felt I was wasting my points in studying science. And at 16, I stood my ground, and I came in to study science, and it was absolutely the right decision. I knew I loved chemistry, and I knew it was what I wanted to do. So one of the pieces of advice I'd give people at that age is, do what you love, right? I mean, there's no question about it. I spend all my days working at something that I love, but it's my hobby, it's not work. And pursuing something that you love rather than what other people want you to do is really, really important. And I suppose the other thing I've learned along the way is the importance of interacting with people. Success in research isn't a solo run. It's all about linking in with people, developing relationships. And sometimes you meet people and they can become important to you 10 or 15 years later. So keeping those links and learning as much about people, and reaching out, taking every opportunity which is offered to you, because there's lots of opportunities out there. It's about seizing them and making the most of them. And as I've shown you along my journey, sometimes I was lucky, things just happened to come together, but it's about seeing the opportunities and being ready to take them when they're offered.
That's absolutely fantastic advice. I'd say everyone here will definitely take that on board. So thank you very much for all that advice. So if there's no other questions, I'll conclude things there. So on behalf of UCC Science Society and everyone here tonight, I would like to thank you sincerely very much, Professor Maguire, for a fantastic and insightful STEMinar. You've certainly succeeded in inspiring us all and have provided us with very fascinating and beneficial advice. And just a big thank you for sharing your career, research and insights with us all.